Okay, so I'd like to welcome everyone to the final speaker for our speaker series. This is the fifth this fall, and we have the last one, which is going to be an outstanding one. This is John Carpten. He has recently moved institutions, and he currently serves as the Chief Scientific Officer for the City of Hope, where he also serves as the new Director of the Comprehensive Cancer Center and Director of the Beckman Research Institute. We thank John for taking the time out of his busy schedule. He's made a move and he's super busy, and so it's nice that he took the time to come and speak to us this afternoon. Prior to his current appointment, he served as Professor and Chair of the Department of Translational Genomics at the Keck School of Medicine, the University of Southern California, Los Angeles. And before that, he was a Professor and Director of Basic Sciences at Translational Genomics Research Institute, TGen, in Phoenix, Arizona. He attended Lane College, a historically Black college in Jackson, Tennessee, where he completed his degree in biology and received his PhD in molecular and cellular biology from Ohio State University. He did a postdoctoral fellowship at NHGRI, and then he was promoted to tenure track at NHGRI. Dr. Carpten's expertise spans a broad range of disciplines and expertise around genomics, tumor profiling, cancer cell biology, and generally precision genomics. The primary goal is to discover, similar to many of us here, molecular alterations in cancer and how to translate these findings into prevention, diagnosis, and treatment. And in support of that, he develops cutting-edge technologies and novel bioinformatics approaches in his research. And much of his research has really impacted our understanding of a number of cancer types across cancer types. I am particularly interested because earlier this year, John had a paper actually in triple-negative breast cancer where he did spatial transcriptomics, did some novel algorithms to understand cellular interactions in spatial transcriptomics, and within that population-based cohort found by race stratification that there are race-associated changes in hypoxia and then also in immune infiltration. And so obviously super exciting. I mean, something that's, I think, exciting to all of us. John has been honored with numerous awards. I won't list all of them, but I would just say he was named a science trailblazer by Spectrum magazine in 2006. In 21, he was inducted into the AACR Fellows of the Academy and also became a member of the AACR Board of Directors. He is also currently serves as a member of the National Cancer Institute Board of Scientific Counselors, and in 2021 received a presidential appointment to serve as chair of the National Cancer Advisory Board. Fantastic awards for a fantastic speaker. Dr. Carpten, we're very happy to have you here, and thank you for your time, and we look forward to your talk.
Yeah, thanks, Adrian. Hopefully you can hear me okay?
Yep.
Okay, great. Well, thanks for the opportunity to present. University of Pittsburgh, amazing organization, amazing institution, and I'm honored to have been selected as part of your seminar series, so thank you. As a person who has a huge executive administrative load these days, it's always good to take some time away and talk a little bit about science. Today's title is, I think it's a little different from the one I sent, but it's along the same lines, population heterogeneity and cancer genomics. And I should have put in implications for precision oncology. And hopefully as I sort of walk along this presentation, you'll understand why I would say that. I think, so hopefully, there'll be some takeaway points that, cancer disparities are real. They're caused by a multitude of factors. We haven't spent a lot of time, I think, trying to explore potential roles of biology. And in order to do that, we need to take an intentional approach to increase representation in our translational, basic and translational research studies. So these are my disclosures. I think, I typically start these presentations off by, making sure I'm respectful to, all populations and groups. And, so the focus of this presentation will, on cancer disparities, will focus mostly on racial and ethnic underserved groups. But we don't ignore the fact that there are other important diseases and also other important medically underserved communities and populations. But we're going to focus on racial and ethnic cancer disparities in this presentation. Again, this whole concept of heterogeneity, how our limited understanding of both population and cancer heterogeneity can blur important insights into cancer development and sort of the sort of broader complexity of cancer, which limit optimal and more effective approaches for disease management for all people. So cancer disparities, again, there's been a lot of work over the last, I'd say 4 or 5 decades, really focusing on epidemiological data that shows and confirms that there are significant differences, both as we think about cancer incidence, prevalence, as well as mortality rates when we stratify individuals, particularly by self-identified race. The ACS really has taken a lead in generating every year a report, cancer statistics, it's an amazing document. So Cancer Facts and Figures, if you have never seen it or utilized it, the guy Jamal, he's like one of my heroes. He pulls these reports together every year, and I would say about 10 years or so ago, they started, so there was an overall Cancer Facts and Figures report, and they started breaking them out also by race. So now there's one, not only the overall, but there's one for Black African Americans, there's one for Hispanic Latinos, there's one for Asian Pacific Islanders and other groups. And within the Black African American Cancer Facts and Figures document, they typically provide these tables that are essentially the reporting on the calculation of these, what they call the rate ratio. So looking at the rate of a cancer in Blacks versus whites, and calculating the absolute difference, and then showing the actual ratio of the difference. And here we can if you sort the table again by highest rate ratio, you begin to identify the cancer types that have the highest potential or likely disparities. Again, this table on the left is incidence, the table on the right is mortality rates. We can see Kaposi sarcoma, which is an age-related malignancy, both in men and women. And then two rare forms of cancer, myeloma and gastric cancer, predominate or represent some of the more significant disparities in terms of differences in incidence rate. Liver, cholangiocarcinoma, prostate in men, uterine cancer in women. And when we look at death rates, we see prostate cancer percolate up towards the top in men, along with, again, gastric cancer, throat cancer, and myeloma. And then in women, we see gastric, myeloma, cervical, and uterine cancer. So these are some cancers that show significant disparities between the Black and white populations here in the States. Now, we also know that these differences are likely caused by or likely the result of a number of factors and influences. And we have typically separated them into 2 main groups, social or societal factors and biological or genetic factors. And considering the social and societal, we think about the new sort of context of the social determinants of health, the phrase that we are all hearing more and more about, access to health, socioeconomics, financial toxicities, insurance and reimbursement, geocoding, redlining, all of the social factors that likely influence these differences in race, but particularly in outcomes. And then there's also the biological and genetic, and I think that there's been a lot of work that probably started, I would say, back in the mid to late 90s, to try to begin to look at, either germline genetic factors or more recently tumor factors that might show differences when we look at individuals across various races and ethnicities. And a lot of this work has taken place in cancer. And cancer actually may be the area where we've seen the most work in this area. I think it's important to state that these 2 things are probably not happening in a vacuum or independently. We know that there are certain communities, for instance, that are enriched for individuals from, that have been sort of labeled as a specific racial or ethnic group. And interestingly, many of the individuals within those groups share similar genetic ancestral proportions. And so there's the environmental influences, the racial and ethnic and social influences, and particular and possible particular biological or genetic influences that are shared that might lead to impact on the host and or tumor biology, and overall impacting prevalence or disease risk and outcomes. So there's been a significant historical focus on social factors that influence disparities. I remember a lot of the, when I was back during my time as a postdoc, when I really became more interested in studying disparities, most of the conferences and meetings about disparities were focused on social factors, whether they were at the NIH through the Office or Center for Minority Health, or some of the conferences that Lovell Jones was putting on at MD Anderson, the intercontinental conferences, and that Lovell was leading. And there was very little work or investigation looking at genetic or molecular factors that might influence these disparities. And so there's definitely been a lack of investigation, right, and looking at the biology. And then finally, exploring the role of biological differences in tumors across populations could actually help to broaden our understanding of the complexity of cancer and a way to best approach disease management for more effectively for all patients. And I think it's important here that it's not just about trying to increase representation to focus on the disparities, although that, of course, is one of the more important components or hopeful outcomes of our work. But it also adds to understanding of the broader complexity, and I think that's sometimes where we lose that part of the importance and the impact of this work. We sometimes think that when there's for instance with a grant in RFA, it says we would like to emphasize, right, recruitment or involvement of individuals from diverse populations. It's not always just about focusing on those populations, but it's helping to understand the broader complexity of cancer. So as we all know, the TCGA was this massive study, the Cancer Genome Atlas. I think 11, 12, 13,000 tumors were profiled by various comprehensive molecular technologies, whole exomes, RNA-seq, methylation, proteomics analysis across 33 different tumor types. But analysis of those cohorts has shown that roughly less than 10% across most tumor types were from individuals from underrepresented groups. And this included tumor types that represent known and significant cancer disparities: prostate cancer, triple negative, colorectal, endometrial, pediatric leukemia, and myeloma. And these two were actually the focus of studies outside of the TCGA. The MMRF did the large COMPASS study, which I had the privilege to be the initial PI for. And then the childhood genomics consortiums that did a lot of the work in the pediatric cancers. But I think it's not important, again, just from the standpoint of, why do we want to look at these tumors from individuals from underrepresented populations? Well, there is sort of scientific and statistical context here, as well as been shown by a series of studies, that we lack the ability to have power to detect mutations at significant frequencies. And even when we think about tumors from whites, try to have statistical significance or achieve significance around mutations that have frequencies at 5% or below, we don't have power for some important tumor types. And we definitely don't have power for the vast majority of tumor types when we look at populations, other populations like Black, African Americans, Hispanic, Latinos, Asians, American Indians, and the like. So this, from our perspective, remains an important unmet need in helping to determine if these genomic differences are associated with some of the disparities that we see in incidence and outcomes. Again, I mentioned TCGA did have limited representation, but individuals have actually taken that data and performed, analyses where they stratified individuals by population. And they did it in two ways. They did it by, the self-identified race information that they had on the patients, but they also used genetic ancestry. So they were able to take whole exome sequencing data from the germlines of those patients and deduce genetic ancestral proportions for each individual and look at the somatic mutation profiles and begin to ask questions about differences in mutation frequency by race and or ancestry. And there were two studies that utilized the TCGA data set, and they showed that tumors from African Americans had higher levels of chromosomal instability for these solid tumors. They also showed higher P53 mutation frequencies and amplification at the cyclin E locus, a decreased prevalence of VHL and PBRM1 mutations in renal cancers. They also showed, which has I think been revealed before, which is a lower overall frequency of alterations that are part of the PI3 kinase AKT pathway, PIK3CA, AKT, and PTEN. And increased FBXW7 mutations in patients of African origin. So again, even though there was limited overall representation in TCGA, just looking at that data because of the large size, some differences across racial groups. So what I wanted to you specific examples and some vignettes as we think about specific tumor types and work that has been done in analyzing diverse populations and sort of showing these differences, and then maybe towards the end get into a little bit of why this is important as we consider aspects related to precision medicine and precision oncology. So prostate cancer, as we know, is the most common male-specific cancer. There's been this historical disparity among Black men, where Black men tend to have significantly higher incidence and or death rates from prostate cancer. And even though we've seen over time a trend, right, of reduced incidence and mortality, we still see this significant gap where Black men are impacted by this disease at much higher rate. So there's two different contexts I want to look at here. One is germline genetics, so inherited genetics and risk. And so many of you know that in the early 2000s, we were sort of in the throes of the GWAS era, the genome-wide association studies, and we're looking for these common genetic variants that might impart increased risk for various common complex diseases, heart disease, diabetes, hypertension, but also cancer. And over time, a number of these studies began to identify these SNPs, or these genetic variants that reached genome-wide significance, where the presence of that genomic or that genetic risk factor would influence increased risk for a particular disease, in this case, prostate cancer. And over time, we began to see more studies focused on individuals from different populations. And of course, because of this significant disparity in prostate cancer among Black men, some of the larger GWAS in African Americans were performed around prostate cancer in Black men. And this is one of those studies. Chris Heyman was actually co-author on the study, where we looked at a series of genetic variants and almost 5,000 African American men with prostate cancer and roughly 5,000 controls. And there'd been a series of genetic variants, risk variants, that had been identified for prostate cancer. And of interest is a series of variants that map to a specific region of the genome, 8q24. And many of the genetic variants that increase risk for prostate cancer, even in men of European descent, reside within that region on 8q24. And what's really interesting about this is that many of the genetic variants that map to that region that have that show or impart significant increase in risk are enriched in populations from sub-Saharan West Africa. So whether we're looking at prostate cancer or not, if we just look at population genetics, these genetic variants that significantly increase risk for prostate cancer are enriched in those populations. So it's believed that there's a potential genetic link, or the fact that these genetic markers are enriched in sub-Saharan West African populations, and that gene flow during the transatlantic slave trade might be at least in part associated with the increased risk in prostate cancer incidence rates among Black men here in the States. And then transitioning to sort of the somatic, so the tumor biology. I'd mentioned the Cancer Genome Atlas, and one of the studies they performed was a study to look at the somatic framework of prostate tumors, where they revealed the molecular taxonomy of primary prostate cancer, and for the first time molecularly subtyped prostate cancer based on specific molecular events that occur in the tumors. And some of the more common events, including the ERG translocations and fusions across the ERG family members, ERG, ETV1, ETV4, and FLT1, and translocations with other genes that encode proteins that are driven by androgen signaling, SPOP, FOXA1 mutations, IDH1 mutations, and other more rare mutations. But they began to subdivide prostate cancer molecularly for the first time. But interestingly, of the 330 or so patients, tumors that were profiled, only about 25 were derived from men who self-identified as African American. So even though we have this disease that has a significant and historical disparity, there was a significant underrepresentation of tumors from Black men. So what do we do? So two of my dear friends and colleagues, Levi Garraway and Ike Powell. This was when Levi was still at the Broad Institute. Ike Powell, who's a really amazing urologist, who was at Wayne State, actually performed a deep whole exome sequencing study to look for mutations in tumors derived from Black men. And they profiled, I think, roughly 100 tumors, all derived from men who self-identified as African American, and actually identified mutations in a gene called ERF for the first time that wasn't identified or highlighted through the TCGA studies. And they were able to show that mutations are highly enriched, if I wouldn't say specific to Black men, but highly enriched in tumors derived from Black men. They went on to do some in vitro studies to show that if you knock this gene out, it increases clonogenic growth in LN-CAP cells in an androgen-dependent manner. And Charles Sawyers and others sort of published a review article around alterations within the ETS family, and that these ERF mutations reveal sort of a different way of getting at ETS transcription factor activation in prostate tumors. But again, with this specific alteration being increased in tumors derived from Black men. I think there's the other important, I think, somatic context is the transcriptional context. And I think it's really important in prostate cancer, not just from sort of a understanding the biology standpoint, but also from a clinical standpoint. So a number of companies have developed and provide clinical tests that can provide physicians with information on whether cancer might be more aggressive or might be associated with increased response to specific therapies. So Decipher, Polaris, Oncotype DX, prostate cancer. So these tests, interestingly, were developed by profiling a large set of tumors where deep and comprehensive and accurate outcomes data was available. So there's a discovery phase, of course, to identify the genes that are differentially expressed in tumors, for instance, that are associated with aggressive versus non-aggressive disease. And then a large validation study will follow so that the clinical validation can be completed for these tests to become available as actual clinical tests, or be developed and credentialed as actual clinical tests. However, one issue is that the vast majority of these data sets from which these signatures were derived were from largely European descent cohorts. For instance, I know one was a large cohort from the Mayo Clinic in Rochester, New York, and there's very, very little diversity and representation in the patient cohort. So we have these tests that were derived from tumors that came from one specific population group. And so, of course, the question is that one would ask is, will the performance men from various ethnic or racial groups? And it's interesting in that a number of gene expression studies have been published, with some of the early hints being provided by Stefan Ons at the NCI, Renee Reams at FAMU, Ike Powell, who all independently showed when you look at the gene expression signature associated with inflammatory signals in tumors derived from Black men rather than more oncogenic signals. This was validated more recently by Walter Rayford and Ashutosh Tiwari from gene expression analysis on over 1,000 tumors from Black and white men, and showed that there were distinct transcriptional profiles. Here you can see this plot looking at odds ratios. We have more pathways that are enriched in tumors from Black men versus white men. When you look at the pathways that are enriched in tumors from Black men, we see pathways associated with interferon signaling, TNF alpha signaling. So these inflammatory signatures being enriched in tumors derived from Black men, and also being associated with advanced pathological features, as opposed to tumors derived from white men that tend to be enriched for oncogenic signals like MYC, and other signatures like DNA repair signatures. So these data, I think, taken together, suggest that there are differences potentially in the molecular transcriptional framework of tumors derived from Black versus white men, and they may have not only important biological implications, but also possible clinical implications. And then all of the work Steve Paterno and all of his colleagues have done over the last decade or so, and not only looking at gene expression, but looking at splicing and showing that there's maybe a higher degree of differential splicing in tumors derived from Black men versus white men, and also that some of these differential splicing events are occurring in important oncogenic signaling and immunological signaling pathways. And I know Steve and his team actually are working on developing drugs to target specific isoforms of proteins by where that isoform is enriched in tumors from Black men because of differential splicing. And so again, there remains this question of the generalizability of some of these currently available gene expression clinical tests. Will the performance generalize, right? In a similar way in tumors derived from white versus Black men. And so we have to assess these in large cohorts of Black or African American men to understand the true effectiveness and accuracy for these tests across populations. And I know some of this work is underway. I know Ike Powell has been working with some of these groups, and so has Walter Rayford. So I think switching gears a little bit to breast cancer. Breast cancer has a little bit of a different, I think, epidemiological signature when we look at at the cancers in Black versus white women, where the incidence rates are higher in white women. However, mortality rates are higher in Black women. And particularly when we look at young premenopausal Black women. And these data have been validated even in sub-Saharan West Africa. I know Lisa Newman has done a lot of work in Ghana and other areas of Africa. And in some of these areas, these triple negative or basal-like breast cancers tend to be enriched in young premenopausal Black women and could be associated with the increased death rates that we see among Black women. And these rates may be as high as 80% in women in sub-Saharan West Africa. So the general frequency of triple negative breast cancer is about, I think, let's say 15 to 20% of diagnoses in white women, probably 30 or 40 in Africa, young premenopausal African American women, but upwards of 80% in women from certain regions of sub-Saharan West Africa. A number of individuals have been, very involved in this work. Chuck Perot and Fumio Lapati have pioneered and spearheaded a lot of this work. I mentioned Lisa Newman, Melissa Davis, and a number of individuals have really been focusing on looking at the differences in the molecular framework of tumors, breast tumors from Black, African American, and as compared to European descent cases. And so Fumi and Chuck performed a study where they looked at 900 patients with breast cancer. It included about 150 tumors or 150 or so Black patients, and showed that they had, Black patients had worse breast cancer-free interval and higher likelihood of basal-like and HER2 breast cancer, as I've mentioned. They also went ahead and looked at some of the common somatic alterations that are known to occur in breast cancer and showed that a specific gene, CRYBB2, the expression tended to be higher in tumors derived from Black women. They also looked at the germline and showed that when you looked at a polygenic risk score based on a series of genetic variants that are known to increase risk for breast cancer, that when you looked at breast cancer as a whole, there really wasn't a general difference in the polygenic risk scores. Then when you stratified by hormone receptor positive versus hormone receptor negative or ER negative, the triple negative, particularly that group, but also the HER2s, we can see that there was an increase in the polygenic risk scores in tumors derived from Black women or Black breast cancer cases, suggesting that there could be possibly a genetic influence when we think about the ER or hormone receptor negative breast cancers that are more common in Black women. Fumi Olopati also performed and presented the results on a study where she characterized breast cancers from women from Nigeria. And it was really interesting. So there were almost 200 cases in this study. And relative to Black and white cohorts in the TCGA, Nigerian ER HER2 tumors seem to be characterized by increased homologous recombination deficiency, P53 mutation, and greater structural variation, indicating aggressive biology. And these, interestingly, are features associated with triple negative. So these were tumors that were actually hormone receptor positive, but they had profiles that were more similar to hormone receptor negative tumors. They also showed enrichment of GATA3 mutations in these tumors derived from Nigerian women. I'd mentioned Lisa Newman and Melissa Davis, who've created the International Center for the Study of Breast Cancer Subtypes, and they do an extensive amount of work across the African diaspora here in the States and the Caribbean, but also in a number of regions in Africa that span across Ghana, Uganda, Kenya, and all the way over to Ethiopia and East Africa to look at similarities or differences in the molecular features of tumors derived from women across the African diaspora, and looking at genetic ancestry and asking questions about the influence of African ancestry, sub-Saharan West African ancestry, on the biology of tumors. They've published a number of studies. Melissa more recently had an amazing paper published in Cancer in Cancer Discovery, where she profiled several hundred tumors derived from Black women from the States as well as various regions in Africa, and identified interesting correlations between the genetic ancestry, the oncogenic signals, and the immunological features of these tumors based on genetic ancestral proportions. As Adrian mentioned, my laboratory more recently has been focused on the utilization of advanced molecular technologies. And this particular example, we published a paper, and it was on the cover of Cancer Research earlier this year, where we applied spatial transcriptomic technologies to assess the tumor and immune microenvironment of triple negative breast cancers. We had a total of 30 or so sections that we assessed, half of which were derived from women who self-identified as Black African American, the other half derived from women who self-identified as European American, utilizing these technologies again to look across the spatial architecture of triple negative breast cancer from a transcriptomic perspective. and actually were able to detect specific regions and molecular features and pathways that were enriched in specific regions of these tumors. As Adrian mentioned, we were able to perform a gene set enrichment analysis on the clusters that were sort of associated with various regions on these sections, to basically take that information and ask questions about the feature profiles, and we're able to show that one specific pathway and cluster, one specific cluster, cluster 5, which was enriched for the hypoxic transcriptional pathway, was more common or more enriched in tumor sections derived from Black women. We were able to validate that in an independent set of cases, which would suggest that there's a higher or an enrichment of cells that are in a hypoxic state in tumors derived from Black women. And that could have implications on tumor aggressiveness, tumor growth, as well as response to therapy and immune response. And that cells that are growing in a hypoxic region are growing away from the blood vessels, so they're going to be farther away from diffusion of chemotherapies. They're going to be farther away from the influx of immune effector cells into the tumor microenvironment. And so we're really excited about these results and are in the throes of finalizing an R01 to validate these in vitro and in vivo, as well as hopefully clinically. So also want to walk through a few additional examples. Multiple myeloma, which is a cancer of the bone marrow. It's a rarer form of cancer. There have been incredible therapeutic advances for the treatment of myeloma. I began working in myeloma back in 2003, and over the last 20 years, the survival rates have trended upwards in an unbelievable fashion. I would say back then, the 5-year survival rates were in the roughly around 30%. Now they're above 50%. There's a very well-known pathogenesis for myeloma where the translocations at the IgH locus occur in B cells in the germinal center, which advance onto monoclonal gammopathy of undetermined significance, which is sort of a pre-malignant state. And many individuals in the general population will have MGUS, but some of those MGUS will go on to advance into what we call smoldering or intramedullary myeloma, and then on to full-blown myeloma and plasma cell leukemias. And there's also a very well-known molecular pathogenesis that occurs along this trajectory. And as mentioned, the 5-year survival rates have gone up substantially because of advances in therapeutics, particularly the immunomodulatory agents like lenalidomide and the proteasome inhibitors like carfilzomab and bortezomib. As mentioned earlier, when we look at the rate ratios, myeloma represents one of the more significant disparities, both from the standpoint of incidence and death rates similar to prostate cancer. I was able to lead and participate in one of the largest molecular studies performed, which was a study called COMPASS that was funded by the Multiple Myeloma Research Foundation, or MMRF, where there was several unique aspects of this study compared to other large cancer genomic studies, in that we targeted over 1,000 patients, but the patients all had to be treatment naive, meaning these were all newly diagnosed treatment naive patients, and all of the patients were followed longitudinally for upwards of 10 years. And then finally, all of the patients would have been likely on one of two regimens, either a immunomodulatory regimen or proteasome regimen, or a combination. And as we advanced through the profiling of these patients, where bone marrow aspirates were collected, myeloma cells were purified, blood samples were also collected for germline, we actually were depositing interim analyses of data. So this was whole exome RNA sequencing and shallow whole genome. And by the time we got to about 700 patients or so, we realized that we had an interesting cohort in our hands by where we had over 100 tumors that were derived from African-American patients. that would allow us to begin to compare the molecular somatic profiles of tumors, myeloma tumors derived from Black versus white patients, to determine whether or not there were any differences that might be associated with the disparities that we see. Again, for the first time, providing a very, very comprehensive picture of the somatic compendium of somatic alterations in myelomas, identifying many of the commonly known mutations like K and NRAS, FAM46C, RYR1, et cetera. But we also, again, had the opportunity to stratify patients by race, by genetic ancestry, and then look at mutation frequencies as a function of race and or ancestry. And for the first time, identified genes that had mutations that were higher or more frequent in tumors derived from Blacks. But what really jumped out or stood out to us was P53, where we saw mutations almost exclusively in tumors derived from whites, with a seven-fold increase in P50, which showed a seven-fold increase in P53 mutation. And looking at genetic ancestry, wild type P53 versus mutant, we could see that the vast majority of tumors that harbored P53 mutations were derived from individuals with higher European ancestry. And even the two African Americans that had individuals who self-identified as African American that harbored whose tumors harbored P53 mutations, they had higher proportions of European ancestry, as we get up to 30–35% in those individuals. And so these data for the first time sort of allowed us to look at the similarities or differences in the molecular framework of tumors, myelomas derived from Black versus white patients. And again, the P53 result jumped out or stood out to us because P53 loss is an independent prognosticator of poor outcome in myeloma. So it sort of went against the dogma to some degree, which would suggest that maybe African Americans have tumors associated with more favorable outcomes. And if we can get them equal access to equal treatment, they may do as well as their European counterparts. And based on a number of studies that utilize real-world data, that's exactly what we're seeing. So this is a publication that showed that when they looked at patients that had been treated with immunomodulatory agents and proteasome inhibitors and stratified by race, they saw that the overall survival for increased to 52% for Blacks and 50% for whites. So we're now beginning to see that we can eliminate a disparity in outcome if we can actually get individuals the appropriate therapies that they need, regardless of race or ethnicity. And this is just another study that also showed that when patients given these blockbuster drugs, when you stratify, we see increased survival probability in Blacks overall, as well as when we're looking particularly at the younger patients diagnosed at ages 65 or earlier. Now, this is interesting that although ours and other data are supporting a socioeconomic access issue that's related to the myeloma disparities and outcomes, it does not explain the persisting disparity in incidence rate. So a number of studies are underway to look at sort of the conversion rate of MGUS to myeloma in Blacks versus whites. Are there immunological factors, germline genetic risk factors, environmental exposures, or some combination of the above that are associated with this significant disparity in incidence rate or disease risk in Blacks that we see in myeloma. And just two more examples, colorectal cancer. I'll walk through these a little faster. So in collaboration with Xavier Lohr and Nathan Ellis, we published a study looking at the genomic or somatic mutation profiles in colorectal cancers derived from Black versus white patients. We profiled 45 tumors from Blacks and then compared these to results from the TCGA. And for the first time, sort of provided a framework of the somatic mutation profile in colorectal cancers from African American patients, again, comparing those to TCGA. One of the things that jumped out at us was that there were a subset of patients that did not harbor APC mutations, and we saw a difference in the APC frequency in tumors derived from Blacks versus whites. And we were able to show that these APC mutation negative patients tended to be diagnosed at an earlier age and had a lower overall tumor mutation burden. We also were able to look at these particular patients and these tumors that were APC negative versus positive, and they also showed distinct epigenetic profiles as well. So this was published a couple of years ago. Now with some of the larger data sets like Project Genie and some of the other larger targeted data sets, individuals are looking at these differences and trying to validate them. And then my final example is lung cancer. Incredible study out of Matt Meyerson's lab at the Broad Institute, where they looked at lung cancers derived from individuals across the Latin diaspora, and were able to show that there was a higher frequency of EGFR mutations compared to Black and white individuals here in the States. So as we think about EGFR mutations in Blacks and whites, there are roughly 12% across those two populations. And when we look at individuals from Mexico, Colombia, and other areas in South America, we see a significantly higher rate of EGFR mutations. And interestingly, it's somewhere between what we see in Southeast Asia, where we see even higher rates of EGFR mutations. And that may explain to some degree the potential migration of individuals out of Asia across the Bering Strait down into the Americas, where we can see this higher overall rate of EGFR mutations in individuals, Mesoamerican individuals, but so higher than Blacks and whites, but lower than Asians. And this could, of course, have significant translational impact, as we know that targeting EGFR is a very prominent therapeutic approach for the treatment of non-small cell lung cancer. And so I think, the culmination of this work sort of speaks to, a concept that I actually wrote a chapter on for the ACR Inaugural Progress Report on Cancer Disparities, a chapter that Shanita Hughes-Halbert and I wrote entitled The Imprecision of Precision Medicine. And so these precision approaches sort of, being defined as a more personalized approach to clinical management, taking into account genes, proteins, environment, diagnosis, and treatment. However, we have a limited knowledge, right, of the cancer biology as we look across racial and ethnic groups. And this, to some degree, diminishes the potential for precision medicine from our standpoint. And it's not just from the somatic aspect of identifying mutations and aligning those to targeted therapies, but also the example I showed in prostate cancer with the gene expression clinical tests that are being utilized, because that also could be construed as a precision approach to cancer management. So we've got to do better in enriching and increasing representation in these molecular studies in order for us to increase the number of data points. When we think about precision is about data points. The more data points you have, the more accurate you can be. The fewer data points, the less accurate you're likely to be. And so by missing many of these data points, I'll use it as an opportunity, right, for us to increase representation so we can fill in those gaps and be more precise in our approaches to manage disease. And then finally, I think as we consider precision medicine, we have to think about it, again, beyond just the genetics and the genomics, but also as we think about the social factors, the environmental influences, and thinking about these more multi-disciplinary approaches as we think about cancer disparities and collecting all of the relevant information and data to, again, be more as precise as we possibly can to manage disease. As we consider the environmental exposures, social stressors and conditions, the built environment, and the impact of these environmental exposures on physiology, epigenetic changes, transcriptional changes, protein modification, lipid metabolism, chronic inflammation, right? And how these physiologies impact cellular biology and lead to pathways, the deregulation of pathways that are associated with the hallmarks of cancer. So have been engaged in a really large national population-based cohort study for prostate cancer called Respond, to define the roles of genetics, immunity, access to care, and other social factors across 10,000 African-American men with incident prostate cancer. We're very well along the throes of this study. We've completed whole exome sequencing on about 250 aggressive prostate cancer cases. RNA sequencing is underway. Deep immunological proteomics analysis is also underway. We're performing whole exome sequencing on the germ lines of all 10,000 men, and we're collecting deep epidemiological and social information on all of these men so that we can begin to build these comprehensive frameworks for what might be driving advanced aggressive prostate cancer in Black men. So in summary, health disparities are real, likely caused by a variety of factors. Differences in tumor biology are not well understood due to the significant dearth of data derived from diverse patient populations. There's mounting evidence, however, from recent genomic studies demonstrating that these differences do exist. And the profiles are not likely going to always be generalizable. Uncovering these factors may help us better understand disease etiologies within different populations, and could provide unique opportunities for improving disease management. So we have to ensure that patient diversity and representation in discovering validation studies and in clinical translational research to increase our ability to be more thoughtful and more precise in our approaches, particularly as we consider precision medicine, where the promise is to deliver the most appropriate therapeutic management to each patient's cancer, again, based on a combination of features. But there's challenges do persist, as we still have a long way to go. A number of considerations and additional obstacles remain to be addressed, including aspects related to population and tumor heterogeneity. Also, will the approaches lead to improved outcomes and effectiveness without sacrificing efficiencies to the healthcare system, and also not exacerbating disparities through lack of access to these innovations? And so at the end of the day, thus making sure that these technologies, if successful and impactful, are affordable and distributed equitably to all people. So I think that's my last slide. Yep, thanks.
Fantastic. Thank you, John. I will clap on behalf of the whole audience. It was that was really fantastic. You took a complex subject and distilled it, and I think it was great that you gave specific examples rather than staying at the high level. You actually drilled down to the true examples. If people want to ask questions, they can put them in the chat. And John, I'm going to start with the first one. Do you want to stop screen sharing?
Yeah, sure.
Yeah, that'd be good. Yeah, that's great. I was fortunate last month to hear Otis Brawley speak. And he mentioned, he's obviously, as you know, a great speaker, and he mentioned that AQ24 that you mentioned in prostate cancer. And I think he was suggesting that there are actually, chunks of AQ24 that are moving around, and, even those regions of the chromosomes if found in white men actually increase their risk as well. Yeah,
absolutely.
And so many of the studies, like you pointed out, are GWAS studies on SNPs, because SNPs are easy to measure. They actually are easy to associate with race and genetics. But I wonder if there have been studies on DNA structural variants, more duplications, large-scale rearrangements, those sorts of things, which are understudied because they're harder to look at. Not now with single molecule sequencing, it's going to change. But I wonder if that, presumably that's a fertile area for research as well, yeah?
I agree. There have been a few individuals who have attempted Diptosri Mandel at LSU. I know is one of the people who was trying to, sort of approach this, but I think you hit the nail on the head. It's the technology and our inability, right, to accurately and appropriately measure them, right? But we know that they do exist. I have not seen a large number of specific examples, right, where we see significant differences. But I believe that, now that whole genome sequencing is becoming more affordable, and also as we begin to move more towards the utilization and application of long read technologies, we'll begin to, I think, dig deeper. And to me, it may not just be it may be the breakpoints, right? And the importance of the breakpoints, not just the structural events themselves, but the actual breakpoints. So I think that it's a fertile area, Adrian, and I think we're going to hear more about it probably in the coming let's say 5 to 10 years as these technologies become more available and more affordable.
Yeah, fantastic. Okay, it's coming up to 5. I do not see any questions in the chat. So if no questions in there, John, I'm going to thank you. I'm sure there's going to be a lot of people that are going to watch this after the fact. I really appreciate you taking the time. I hope your conference goes well on precision oncology this evening.
Yeah,
And
flip me an email if there are any additional questions
yeah,
or anything that anyone.
certainly, I'm going to hit you up for your chapter on the imprecision of precision medicine.
Yeah, you got to check it out.
All right. Thank you, John.
Have a
Bye.
good afternoon. Bye.